Chapter 4, Section 9, Biofilm Growth. Microbes that are growing in suspension, floating around in a liquid, a broth, the water in a stream, whatever. We refer to these as cells being in a state of planktonic growth. Planktonic, okay? Plankton, planktonic. They're free growing, free swimming, free floating. If these microbes have the ability and they attach to something, a surface, and now instead of being free floating, they're now growing, going through binary fission or whatever, while attached to a surface. A rock, the enamel of your teeth, maybe it's, the tissue of one of your heart valves, okay, whatever. This is now referred to as sessile growth. Sessile growth can lead to the development of what is referred to as biofilms. Biofilms can be layers of one bacterial species, that whole colony thing, but now instead of growing, free floating around individual cells, they're all on top of each other and they're on a surface they shouldn't be able to attach to, but they now can. Or it could be multiple species. One species can adhere, other species adheres to that first species. And now you get layers and layers of different bacterial species growing where they shouldn't be able to. When we study biofilms, and biofilms, it turns out, is one of the hot new areas to study in microbiology. We study the bacteria, the archaea that are growing in these biofilms, the fungi and whatnot that are growing in these biofilms. We see that the cells may be the same species, but cells of the same species that are growing planktonically are going to be expressing different genes, different levels of expression, different metabolic activities, metabolic profiles, when they are in this sessile or a biofilm state. These biofilms, as the bacteria begin to adhere, either to the surface themselves or they adhere to other microbes that are adhered to a surface, so they start layering, we begin to see that the polysaccharide matrix, a meshwork, starts to form over all of them, holding them all in place, making a pseudo-bacterial, microbial, biological shield. Let certain things in, keep certain things out. Other microbes attach to this polysaccharide meshwork, and then they build more layers on top of that. So when you see, and you look at this, you study this, you see that it happens in stages. Some planktonic cell that has a flagella, fimbria, pilla. Remember fimbria and pillae? These are great for attachments to things, adherence. They stick to something. You see that once they stick, they start going through binary fission. One stuck becomes two stuck, becomes four stuck. You are now seeing colonization. You'll see extra polysaccharides being made and deposited around the cells that are all clumped together. There's that meshwork from the last slide. As the more and more cells get clumped together and enmeshed, they start to affect gene expression in each other. Epigenetics starts to kick in. High levels of certain metabolic, products, waste products will trigger different gene expression profiles. So you begin to see metabolic changes, gene expression changes. Certain enzymes go away, new enzymes come about. You can see this in this computer-generated microscopic picture series. A few get attached. You see the colonization as those few start to, going through. 1 becomes 2, 4, and 18. And then they start to layer up. You see that polysaccharide enmeshment begin, and more layers, until it hits a certain point where the balance has now been lost. Okay, it's hit this threshold point where there's just too much there, and the gene expression of these bacteria is such that it's going to make them leave. And the biofilm will start breaking apart. Individual planktonic cells will float away. Chunks of the biofilm will float away, disperse to new areas. We'll see this in nature, with the formation of what is referred to as microbial mats. Next time you happen to be out walking around a stream or something like that, you'll see that slime layer that can be on certain rocks. If you get down close and you look at it, that's actually a biofilm of different protists, bacteria, and fungi and algae that are now growing. Something was able to adhere to the rock even though the water is flowing by. And then, 1 became 2, 4, 8, and then more started to adhere to them, and it just keeps going. See this picture there? There's one of those little biomats. This biomat is great for these organisms. Water flowing over brings in nutrients, carries away some of the waste products. But as all these microbes are there together, they are protected. They can somewhat control their little microenvironment here for changes pH, changes in salinity, changes in osmolarity.